There is a bunch of apps here.
I guess that's a good place to stand.
Yeah, it's very good.
Well, good afternoon. It's always such a privilege to have one of these events where a student who's worked very hard over a number of years comes to a culminating experience of providing the community with his wonderful research and discoveries. Amin got his BS degree at the Faculty of Sciences, Rabat, Morocco. Came to the United States. I think he said that his friends used to call him Amin the American.
Yeah.
And came to the United States in 2005. Worked in an immunology lab at the University of Florida before coming up to Pittsburgh and joining the Kagan lab, where he did wonderful things, and then entered the program for the PhD. Amin has been an amazing student, and I say this in the context of more than a dozen students of mine. Each of them is somewhat unique, but I think that Amin has been extremely unique in his intellectual exercise. And if there's anything that a graduate student can take away from what he has done, is to read, because I've been amazed at the depth of his understanding of the literature in many different fields that has really helped to make a strong thesis dissertation. So he's certainly fulfilled the academic portions, but I think beyond that, he's made some novel discoveries that I hope he'll share with you today. And I will allow him to explain the meaning of his title.
Okay, thank you very much. Thank you so much, Dr. Warshawski, and for this nice introduction. And first of all, I would like to start by the most important thing. So I would like just to say that anything good in this dissertation, I owe it to four people. Dr. Warshawski and Dr. Ambrosio, of course, with whom I actually learned a lot, and most of this work is ideas coming from them or through interaction with them, and Dr. Valeria Kagan and Dr. Julia Bayir, because this is the two persons who gave me the opportunity to join the department. He gave me the opportunity to learn, to grow, and to learn how to think. So I'm very, very grateful, and I'm honored by your presence, Dr. Kagan. Okay, so I think there is no better way to introduce my presentation than actually letting Dr. Ambrosio and Dr. Warshawski themselves do that for me.
Literature that has looked at the effect of environmental exposures important for stem cell function into adulthood and how that may affect tissue healing capacity. Across the world, we know that over 140 million individuals are exposed chronically to arsenic in their drinking water supply. Within the United States, we know that there's over 4 million individuals that are chronically exposed to arsenic.
Arsenic is the 21st most abundant element in the Earth's crust, and most of the contamination in drinking water is from the bedrock and wells there. So the experiment of paradigm with mice is essentially just exposing them to arsenic in drinking water as humans would be. We use a level of arsenic
And that's exactly what I was trying to do in my work. So, okay, is this working? Okay.
View ourselves as pioneers.
Okay, so let me just start by just giving some quick background about just some explanation of the title. Okay, so the power law refer to a mathematical law. So this is a distribution basically. So there is the most common distribution we are dealing with is the normal distribution. The power law fall in the realm of extreme value distributions, and it's a phenomenon that is pervasive of many fields in physics, sociology, medicine, biology, oceanology, seismology. The interesting thing about this law is that there is this idea that there might have been or there might be some kind of fundamental mechanism that is universal that can explain to us how complex systems work in general, regardless of the discipline we are dealing with. And as we move forward in the presentation, this idea will become clearer. In terms of optimization, so here I refer to the interplay of all these mechanisms at the organelle level and cellular level and even cell population level that interplay in order to give you a certain outcome, the best outcome possible for the cell population. And the rest of the title, I think it's self-explanatory. So we are talking about cellular memory. That means, in this case, we are exposing our cells to a stressor for a period of time. We are removing that stressor from the system, and then we see the effects being sustained and lasting over a certain generation of time. Okay? So again, what you've just seen in the video is just one facet of the project. In fact, this is a multifaceted project with the different goals. So basically trying to address both the intrinsic cues that may be responsible for altering the regeneration of muscle as well as the extrinsic one. So the matrix will be the extrinsic cues, and in my case, I'm interested more in the intrinsic cues that may actually contribute to the impairment of the muscle regenerative capacity because of arsenic. Okay, I will just give you a quick overview regarding regenerative events. So normally in the muscle, so there is there's a small population of cells, like maybe 2 to 5% of cells. We call them satellite cells, and this is the skeletal muscle stem cell. They're quiescent, okay? And quiescent here mean, in a large sense, it means that they are metabolically quiet. So that's what it means. They are not doing much. But when there is an injury, this quiescent cell will exit their quiescent state, get activated, and start to proliferate. And during the proliferation, there is two possible scenarios. The first one, they may go through a symmetric division and then give you two more cells that may actually move forward to a myocyte state and then fuse and regenerate the damaged muscle. Or they can have an asymmetric division whereby you have a cell that actually go to the ground-state quiescence. And so this is actually one of the most fascinating, actually, or fundamental problem in regenerative medicine and stem cell biology is to understand how this transition actually happened, how they are regulated, how we move from here to here, how we go back, and what define actually its state. And so far, there is actually one big kind of theoretical or conceptual framework that explains that, which is the metaphor that Waddington came up with in the late 50s, which basically tried to explain how the interplay of gene expression, configuration of gene will define a state. And he gave this metaphor with this hill and with marbles moving towards the hill, and each each minima is a state. We'll define a state with a certain configuration of gene expression. But we know a little bit about certain states. So its minima will be some type of cell. He made this metaphor in the context of embryogenesis. So each minima will be a muscle, neuron, or something else. But it's also applied within a specific kind of stem cell line. And, but our understanding is very limited with regard to how these moves come. So we don't know which pathway the cell will take to move from here to here. Does it go this way, this way? We have no idea. And that's also kind of part, is another facet of another problem, which is the debate whether the cell fate is determined by extrinsic cues or intrinsic cues. So people who say extrinsic, there is a lot of experiments that support that, think that the environment dictates the fate. So the environment has some kind of instructive role that dictates on the cell where to go, what to become. And people do very smart experiments to prove that point. On the other hand, there are people who actually don't agree with that and say that the fact that the cell takes a certain fate under certain environmental condition is just a manifestation of some intrinsic feature in the cell per se, and not actually. So you give the cell some environment, it will compute that environment, and it will respond to that environment. But doesn't mean that environment imposed is the cell has something in it that actually responds that way. And there are also, of course, very smart people who take the midway and say it's the interplay of both. And of course, the truth by its very nature is kind of reconciliatory. So yes, so it makes sense to say that there is intrinsic. So then the question, then this become actually obsolete question. This is not the right question to ask. And this is what I have been thinking about. I think the question that we should ask is not whether this is extrinsic or intrinsic, but how extrinsic and intrinsic actually interplay to define the fate. And being a product of Kegel lab, so of course the first thing I think of is mitochondria. So I have been thinking about mitochondria, and mitochondria is a very unique organelle because it has, so maybe I'm going ahead of myself here a little bit. So what I wanted to say here is that we know lots of details about the genome, about the lipidome, about the proteome. We know lots of things about the signaling and all that. So we know a lot about the hardware, but we don't know about the software, what's run the machine, and what is this integrative mechanism that actually gives us the final output. So, and in lipidomic, in particular, there is some great ideas that have been put forward recently, which I will touch on at the later of this presentation, which actually may be some great answer to this problematic. So you can recognize here the equation that was put by Wallace when he put structure plus energy. So Wallace has some, in 2005, Wallace said, well, you know, I believe that the Occidental medicine is kind of failing in dealing with this problem we have. We didn't find any cure for neurodegenerative disease, no cure for cancer, no cure for diabetes 1. And so he said we need to switch, we need to re-question the premise of what we do. And he proposed that we need to take into consideration energy. And of course, the focus on mitochondria starts from that point on. And that was the equation. And that was the equation he put: structure plus energy equal fate, I believe, so fate, yes. So I add the information, because that's actually now what we are starting to learn is that the information is the third component that we need to take into consideration. And there is a lot of work that is done by people who call themselves biosynthetic biologists, so people who do synthetic biology. And they make some really very nice comparison between the cell and computer devices. And so the idea that somehow the focus, as I mentioned before, should be on trying to figure out the software that runs the machine running that, rather than hardware, which is the biochemistry and all that. And so, as I mentioned before, mitochondria has some unique future. Mitochondria has both autonomous and non-autonomous future. So mitochondria actually can, and it's the central sensor of the cell. So mitochondria can pick up information from the environment and also can pick up information from inside the cell. And so it's poised to play that kind of integrative platform that actually at which we can have that interplay of autonomous, non-autonomous kind of cues, and we can have some output. And of course, the links to the way mitochondria can influence the final stage, which is the epigenetic mechanism, which include transcriptional mechanism, post-translational modifications, and other things, or histone modification, for example. We know that mitochondria can influence all that in many ways, in many formal shapes, directly by exporting metabolites inside the nucleus, or indirectly by actually affecting other elements, signal and pathway in the cell. So I believe 4 years ago or so, during my rotation, I participated in a study. The study consists of exposing mice to arsenic for 2 or 5 weeks, and then collecting the muscle cell, the tissue, and also the satellite cells. And I made some measurements at that time. So one of the most important reasons why actually I got interested in this is because of these images that Dr. Barshowski shared with me when I was during my rotation, and in which we could see clearly that mitochondria is being swollen here. And you can see this is satellite cell. This is the control, 2-week exposed and 5-week exposed. So this is satellite cell that have been taken out of the animal and now are being grown in vitro, and they keep this phenotype, okay? The tissue here, we see the same thing. And what we can notice here that there is a profound remodeling of mitochondria in the tissue, and also mitochondria tend to be a little bit elongated or swollen in the arsenic exposed versus control. And my rotation work was to try to, so I suggested to Dr. Barshowski to look at the mitochondrial dynamics, and so we ordered some primers, some antibodies, and I did some measurements. And indeed, I found that there is a significant change in OPA1, which is the inner mitochondrial membrane fusion protein, but it's also very fundamentally adaptative in metabolic adaptation. For example, if you knock out OPA1, the cell cannot do the move from glycolysis to oxidative phosphorylation or vice versa, for example. And also we found other protein, and all of these proteins actually are lethal. And these measurements are present in muscle, but we found the same results in hippocampus, in cortex, in fat, in other tissues. So this was kind of the starting point when you start to try to understand what's going on mitochondrially. So as mentioned before, because of this unique feature of mitochondria, we hypothesize that mitochondria function as an integrator of cell autonomous and non-autonomous, which stands for extrinsic and extrinsic cues that dynamically direct stem cell transition. So, and the emphasis here is in the word dynamic because of mitochondrial morphology and dynamics. So I believe, that's the kind of intuition that we try to follow, is that this dynamic, considering mitochondria's dynamic system, actually poised very well to play a very good role in directing the trajectory of the transition of the cell. So the first aim was to establish an in vitro model and clarify the interplay of mitochondrial morphology and stem cell transition. And that means the first aim, we would like to try to figure out what's going on when moving from here to here. What's happening here? Who are the players involved? And aim 2 was to examine the epigenetic mechanism for arsenic-induced cellular stress memory, which means that we are trying to figure out what happened here. Once it moves to a certain minima, what's going on in there? And so this is my in vitro model. So we take cells, this is a myoblast, mural myoblast, we put them to proliferate near confluence, and then near confluence, sorry, and then we switch them to differentiation media, which is basically we just starve them. We just take away a lot of serum, we just leave a little bit. And once you do that, they start spontaneously to differentiate. So they proliferate a little bit, and then they start to fuse together, and they form these tubes. I'm taking this picture at low density, just on purpose, so you can see the myotubes. But in reality, so you can see actually the reserve cells. But in reality, this is actually there is a whole network of myotubes in there. So what happens is that when you have the in vitro model, this C2C12 cell we used are actually very good model for for studying the regeneration of skeletal muscle because they mimic pretty much every step in that regeneration event. So from the activation, they share the same feature, proliferation, and going back to ground stage or differentiating and making myotubes. And so what we were interested in is actually the reserve cells. So what we do is we differentiate this for 4 days in presence of arsenic or absence of arsenic, of course, and then we isolate these guys, this reserve cell, the anti-fragile cell, and then we analyze them, okay? And then we do some analysis on them, and then the other option is to put them in proliferation in presence or absence of modulation of mitochondrial function, which is XGB. And then see what happens. So the first measurement is basically we're trying to find the baseline, and from 3-4 days later, we're trying to understand whether we are able to reproduce the memory of stress that we saw in the animal model. So the first thing we did, of course, is try to see whether we were resistant. So I used 20 nanomolar of arsenic. This is very low dose. It's 1 dose of arsenic. This is pretty much the EPA level. And it's amazing that actually we were able to produce the same result that people produce with arsenic for regime of several months of exposure in animal models. So anyway, so when we exposed the cells to arsenic, we want to check whether we were able to induce a stress response. And so we measured the glutathione, total protein tiols, and low molecular weight tiols. And as you can see, there is a strong response. So indicating that the cell that dose of arsenic causes some stress, and the cell is trying to respond to that and adapt to it. After that, we wanted to see, to basically do some functional phenotyping of mitochondria. We measured the mitochondrial potential we both with GC1, both with flow cytometry and with the plate. And we find out that there is a high mitochondrial potential. So the mitochondria that was exposed to arsenic is hyperpolarized. And we confirmed that with microplate with a negative controllosis CCCP. We also saw that there is an increase of mitosocs, which is a reaction species coming from mitochondria. Of course, mitosocs is not that great dye. I mean, it's not very specific, but it's a correlate of what's going on as far as oxidative stress. And we also measured mitochondrial mass with mitochondrial deep red, which also has a limitation being dependent on mitochondrial potential. So we confirmed that by measuring actual protein from the membrane. So next we wanted to see what's happening. So we used also non-acridine orange. And non-acridine orange is a dye that is once used with, like at very low dose at 10 nanomolar, for example, you might be able to detect the red fluorescence. And that red fluorescence has been reported in the literature by some French group to be actually, to be the result of dimerization of NaO with some cardiolipin. So it's actually, according to that group, is a direct reading of the cardiolipin amount that you have. And so if that is true, then we saw a huge decrease here in the red. So you see that this population increased a lot, which means there is some oxidation of cardiolipin going on in it. And we measured actually the protein cardiolipin synthase antibody that was given by Jan 5. And when I measured it, to my surprise, I found actually cardiolipin synthase increased. So it looked like if you oxidize some cardiolipin, that's actually trigger the cardiolipin synthase to start to work to try to replace that, maybe. But in any case, I looked at the other protein of the membrane to see mitochondrial protein dynamics, and we saw an increase. So here I'm probing for OPA1 with 2 different gel. So this is 3 to 8%, and this is 4 to 12%. The reason why I use that is just that makes the oligomer a little bit more clear. So so what you can see is that this increase in the amount of OPA1 as well form, and also increase in the oligomer. And when you look at the details, you can see that there is increase in processing, with parallel increase in the long form as well. And then I looked at the two major metalloproteases that are involved in the processing of OPA1, and I found as well that are increased. OMA1 also oligomeric changed. The oligomeric state of OMA1 also changed. I measured also MFN1, MFN2, and they both changed as well. So overall, they see that all the profusion protein were actually increased. So it looks like we are able to reproduce some of the features that we saw in the animal model in terms of fusion and all that. We also looked at the electron microscopy, and with the electron microscopy we confirmed that there is a remodeling of mitochondria and also swollen mitochondria. So this is exactly, we are recapitulating exactly the in vivo model here. But we also notice there is a lot of autophagy going on. And just, this is not our focus, but just, to be complete, we evaluated my autophagy, we used some markers, and we found actually there is an increase in autophagy. And that's something we expect because the transition from quiescence to activated state has been shown to involve a lot of autophagy. So the cell needs a lot of resources, and that's part of the process. And so we conclude that the reserve cells that arise from arsenic-exposed cell cultures have primed mitochondrial functionality and dynamics. And so the next thing, so we would like to understand how this transition happened. And so we took a step back and just to think about the whole system. And let me just take 2 minutes to explain to you this. Okay, so when you have a quiescent stem cell in an environment and there is an injury, and then they get activated, so now you have basically an optimization problem. So the cell need one, to respond to the injury and try to replace the damaged tissue. Number two, they need to, after doing that, they need also to make sure that the reserve is not, the reserve actually is replenished. And number three, there is always apoptosis going on in the system because apoptosis is needed because the products of apoptosis are needed for the cell to fuse. So the apoptotic cell will release phosphatidylserine. That phosphatidylserine is picked up by other cells as a signal to actually fuse and form the myotubes. And so what's going on here is that you have, So when you have this regenerative event, you have a probability of the cell like beta to proliferate and probability to actually differentiate theta here, and another probability 1 minus mu, which is the probability of apoptosis, because to have apoptosis you need to have, so it's like the total probability should be 1. So whatever probability of apoptosis you take out, the remaining will be for proliferation or differentiation. So this is so thinking about it that way, we said, okay, we're gonna look at proliferation and see what's going on in terms of proliferation, and then we're gonna look at the exit from quiescence to the proliferation to the activated state and see what happened again. We did not consider apoptosis. We were isolating only cells that are alive, so we did not focus on apoptosis. Although I believe apoptosis should be playing a role in this system. Okay, so we did measure proliferation by 2 means. The first one, we loaded the cell with this carboxyl fluorescein succinimidyl ester, and the second one with the Ki67. So this one, you just load your cell with the dye, they proliferate. As they proliferate, the dye get diluted. The more diluted the dye, the more proliferative is your cell. And the Ki67 is a marker of proliferation. It's just you just fix your cell and then label them with Ki67. If you have more Ki67, then you have more proliferation. And the two results actually converge to confirm that there is a hyperproliferation going on in primate cells. So at this point, let me just precise that now we are talking about primate reserve cell progeny. So this is cells that we took and we put in proliferation media to simulate activation, and we let them go in absence of the initial trigger, initial stressor, which is arsenic. So there is no arsenic in the system at all. And now we see this behavior independently of the presence of the stressor, which means that this is a cell autonomous kind of mechanism going on here. So we also wanted to see what is the identity of the cell. We used 2 markers, CD34, which is a marker of regenerative capacity in quiescence, as you can see here, so when the cell are quiescent, they express CD34, they are CD34 positive. If there's an injury, they get activated, and they actually lose the CD34, a requirement for them to move to the next step and commit to differentiation. Okay, this process seems to be dependent, to be regulated by Notch1, which is believed to act like a bistable switch. Okay, so it allows CD34 to switch from plus to minus. And actually in a real regenerative event in an animal, so what you can see is that there is a certain spatiotemporal distribution of this CD34, and it seems to increase during the first 3 days and then just get much, much lower. So we measured this notch, we labeled the cell with Notch1 and CID, which means Notch1 had been processed, and CD34. And what we found is that there is an increase in the population of NCID CD34+. So this indicates that majority of cells that are coming from the progeny of reserve cells that have been exposed are actually in activated state in this area. Okay, and of course we measured Notch1 and we confirmed that Notch1 is increased by Western blot. But Western blot also revealed something else, that the processing is different, but also there is some actually also some kind of oligomer in here. And other bands that I mean, there is a lot of processing going on, but we didn't look into that in detail. There is a lot of literature about that, and it's meant to be a very important thing. So the next thing we looked at is caspases. So we were aware that there is a form of caspase activation during the differentiation. There is a massive activation of caspases, and let me tell you something about differentiation. Differentiation and apoptosis share a lot of pathways together. They recruit the same element, the same pathways, and somehow at some point there is some kind of bifurcation when you get differentiation as opposed to apoptosis. And that bifurcation, how it's called, nobody knows how it actually works. It's kind of a near. I like to think of it like a near-death experience. So the cell like to die, and then either differentiate, go back, or actually die. So we know that caspase-3 in particular, CLIF-PAC7, which is the protein that actually defines the stemness of the cell. And we know also that if mitochondria is persistently hyperfused, it promotes the G2M phase, and that can lead to caspase-dependent cell death. Okay, but we didn't see any cell death in our system. Okay, so we wanted to see what's going on here. So we know now that there is some aberrant proliferation going on, and that the cells are stuck in the activation phase, but we don't know what's going on several more. So we decided to look at that. Going backward. And so what we did, since from the previous figures, as you can see, there is very strong correlation between CD34 and Notch1. So we took advantage of CD34 being a labeled antibody, and we can actually conjugate it with some diet, and we can measure cells that are we can do some live cell measurements. We took advantage of CD34 as a correlate, as a representative of this phenotype, and conjugated with different dyes. First of all, we wanted to see whether the same population of cells that have heightened mitochondrial potential, reactive oxygen species, mass, etc., is the same population that has high CD34. And sure enough, that was the case. Actually, I'm not showing that data here. This is actually for proliferation. So I load the cell with the CFSE again, and then I label them with TMRE, and I label them with MTDR. And when I did that, of course, the more proliferative cell, the less dye you have, and you can see that arsenic clearly have more of dilution compared to control. And so that means that the same population that has this hyperproliferative behavior is the population that also have this heightened mitochondrial profile. But when we also conjugated caspase-3 with it, we actually find out that that same population have very high, twice the caspase activity 3/7. So I was not really like I didn't believe it because my understanding, I was always under the impression that Caspase-3 is synonym of that because you have the cytochrome C release and all that. But when I did actually measure, I used alexin 5 and 3E, there is no that. There is very little. This is probably just due to handling. I don't think it's biological at all. Because I actually use only the cells that are alive. I discard any debris or anything in the media. So that means that we have the same hyperfusion, we have the same activation of the caspase, yet we don't have the same outcome, we diverge. In contrast to that paper I referred to, I have actually an activated stem cell. So we wanted to see whether if this is true, then the mitochondria should be hyperfused. Then we did some imaging with the help of Dr. Sankarwa, and we found out that actually, yes, sure enough, the prime cell have a much fused mitochondria than this one. And we did some neat analysis on this. So basically, we took advantage of the graph theory formalism to actually convert the images that we binarized, convert them to a network, and then start to do some mathematics on this network. And what I'm presenting here is one or two analyses that I did. This is a methodology paper that we are hoping to be able to publish in collaboration with Colin, who does also, Colin does his processing with based on NIS elements and C+ language. And I've done this, I wrote some code with Wolfram language, and I've done this. So I think we can merge that and put in some kind of MATLAB package when we can have a kind of streamlined analysis of mitochondria. And so you can see here that based on that formalism, you can see that the network of arsenic is much more developed compared to the network or the control. And then when you count the node as a correlate for fusion, you can see that the number of vertex, which is the nodes, which indicate fusion, is much higher. And the distribution is actually a power law. So that means that if you take this and you put it in log-log scale, it will be almost a line. And actually, that's what I did when I started to calculate the probability of this. You can see this is the control, and this is the arsenic, and you can see that if you truncate this at this point, you will have basically a line here, which is a power law, because this is a log-log scale. So we conclude that reserve cells with prime mitochondria are hyperproliferative and have limited cell renewal capacity. So, I'm sorry. So that means, that the cells that have been exposed to arsenic get activated and get stuck in that activation phase. They cannot move forward and commit to differentiation, but they cannot go back either and self-renew themselves. So we have cells that are in kind of some kind of poised state. So so as general conclusion, we said that arsenic exposure resolved the reserve cells to a poised-like state by promoting aberrant mitochondrial fusion. Moreover, the sustained mitochondrial phenotype and poised-like state in the absence of the stressors implies the presence of cellular memory component to the prime cell behavior. So I think this is clear. We are having cells that keep the same behavior regardless of the presence of the initial stressor. So there is some kind of cellular memory going on. And so we answered the first aim. And the first aim was what's going on in terms of transition. And now we know what's going on. What's going on is that arsenic is compromising both the differentiation step, the commitment to differentiation, and compromising self-renewal. And that's why we have this profile. And so, because of the cellular memory possibility, I start to look at the literature. What I found is that if you express a protein called H2AZ, which is an histone invariant, if you express that in the cell, then you will block the myoblastome differentiation, exactly what I see in my system. And actually you keep them proliferating. Now H2AZ is very cool protein, because not only is it responsible for epigenetic memory, it's responsible for memory period, the high function memory. It was found that if you affect H2AZ by some means, like deletion or mutation and things of that sort, you affect the functional memory of the brain. And there is, you basically affect some connection between the cortex and the hippocampus. So we were excited about this. And so we decided to look a little bit more into it. And what we found that H2AZ is highly correlated with a lot of epigenetic marks. The trimethylate and this lysine 27 and lysine 4, trimethylated lysine 27 and lysine 4, and also with the activity of polymerase RNA 2. And so we also found that it's highly correlated with the CD34, as you can see here. So the cells, these cells are actually pure CD133 and CD34 cells. And so when we looked at CD33, we found that also to be correlated with high reactive oxygen species production, high mitochondrial potential, high oxygen consumption, high mitochondrial mass, and ECAR, which is basically kind of a sign that there is a Warburg effect. So we measured the H2AZ, and sure enough, H2AZ is increased, the expression of H2AZ is increased in our system. And so this is the quantification of it. And then we looked at these two markers, CD34, CD133 again, and we confirmed that they are increased. So we have, it seems to me that this process is actually realized in our experimental model. So then we turn to XGB. So I was exposed to XGB when I was working as technician for Dr. Bayer, whose student, Dr. Jing, used XGB actually to rescue animals from traumatic brain injury. And XGB, of course, because it's brain permeable, so it can access to the brain. And they got very good results, and they were able actually to reverse the cardiolipin oxidation phenotype. So we thought that may be a good thing to use to kind of modulate. So XGB is an electron scavenger, so I think everybody knows that. And so we use XGB to try to see if we can actually rescue the phenotype. And we started to give to proliferate cell in absence of presence of XGB, and we were able to basically rescue all the mitochondrial phenotype, the mitochondrial potential, mitosox, and mitochondrial deep red. And also we looked at the mitochondrial morphology, and we were able actually to establish that as well. Then we wanted to see whether by actually intervening in mitochondria, we were able to affect the identity of the cells and the fate of the cells. So we looked at these two markers, the CD133 and CD34, which are both markers of regenerative potential. And sure enough, we were able actually to reverse the phenotype, as you can see here. So we brought that down, and we brought them both down here. Well, then we decided to look more closely at this at the single cell level. So we wanted to see what's going on in the cell. And so we stained the cell with H2AZ, which is the memory-bearing cell, and a protein, the memory-conferring protein, and EZH2, which is co-localized with H2AZ. And this is a protein that has been suggested to actually control self-renewal, specifically the transcriptional identity of the cell. And so, so you have H2AZ in red, EZH2 in green, and DAPI, of course, and then you have merge. And you can see, just looking at these images, that we were able, just visually, you can see that we were able actually to revert the phenotype. But aesthetically, you can see that there is a very interesting increase of H. So we confirm the Western blot, number one. But number two, regardless of the condition, there is a very high cell-to-cell variability in terms of protein expression. Very high. So this is not like one kind of normal distribution of things, clearly. Although by using just Gaussian statistics, we are able to say, okay, we did something good. We were able to reverse that. By the way, we also were able to reverse the morphometric of the nucleus. Here an area, but also we looked at the length and the width. So it looks like by acting on mitochondria, you can change even the morphology of the nucleus. So being familiar a little bit with the statistics, I knew that, well, after that, of course, I looked at the correlation between EZH2 and H2AZ, and sure enough, so we used the Spearman instead of Pearson, because Spearman is very, can actually refract to outliers, contrary to Pearson. So Pearson, when you do the correlation with Pearson, if there is some outliers, they will affect the results. And when we did that, we were able actually to improve the correlation between H2AZ and EZH2. So after that, realizing that we have a huge variability, so we realized that probably the Gaussian statistics are not working. We cannot describe this data with only the mean and standard deviation. That will be misleading. And you can see that clearly from the QQ plot. The QQ plot is used by statisticians to show if actually you have a normal distribution. And if you have a normal distribution, then your data should be precisely aligned with this theoretical line. So for example, the control should be aligned with the blue. But it's not. What does it mean? It means that this data actually violate the assumptions that we actually should have when we are actually treating any data with the Gaussian statistic. And so that means that there is something going on here. And we did something called kernel density. This is just a non-parametric way of trying to see the distribution of data without assuming any precise distribution. So you don't restrict yourself to any distribution, you just look. And when we did that, what is clear is that regardless of the condition again, and regardless of the parameter you see, there is always a skewed heavy tail to the right. Okay? So that was an indication that we need to start to think in non-Gaussian way. And we looked at the literature, and there is precedence in this. There's including people from Pittsburgh and CMU who actually did use non-Gaussian statistics to deal with stem cells. And so what this means is that the central limit theory, or that actually we use to do the mean and the standard deviation, say that okay, if you have a large number of some sample, then the variation will be around a certain mean, okay? But that law doesn't tell you that everything in that distribution will obey the law. So things that are here may not obey the central limit theorem. Actually, there is another theorem that was specifically designed to deal with this issue, which is called the extreme value theory. So extreme value theory, as the name indicates, deal with extremals, things that are extreme, things that happen, things that you don't expect to happen, but all of a sudden they become high probability and they happen. They may be rare, but they may happen. Now, you will tell me, well, if you have 1 or 2 or 3 cells, who cares? Well, actually, that's not entirely true. So I'll give you one example here. I took these 2 cells for EZH2 in arsenic, red, these 2 cells. And then I took 25 cells here. And I compared their expression level and you can see that when you do Gaussian statistic and you are reporting averages, what you are reporting is the average of 2 cells. Because these 2 cells actually dominate the distribution because they're extremos, because it's a power law. So that's why we call it a power law, because a power law means that you have a distribution and you have a large number of events, and there is a few events that define the whole dynamic of. This is what we call in economy, for example, the 20-80%. 20% of people have the wealth, have the decision, and 80% of people are actually irrelevant because the 20% make the decision for 80%. This is what we also call the 1%. You know, the Wall Street 1%. The 1% decide, and the millions don't matter because that 1% have so much power that it actually affects the distribution. It's the same thing here. That's actually, it's exact same thing. And that's why we have the collapse of the US markets, stock markets in 1998, is because people who deal with these stocks and stuff didn't see that, didn't see the extreme value theory happening. And there's actually a very nice speech of the chairman of Federal Reserve about this, explaining the extreme value theory, how it is important. Anyways, so that's the reason why, in this particular case, using the Gaussian statistic is not is misleading. It doesn't reflect reality, and it will actually we will lose a lot of information if we go that way. Okay, so this is what power law means. So power law, so you have some distribution of some variable, okay, and you have some constant, and you have some variable up to some power, okay. If this power is negative, then how do you know it's power law? When you put it in log-log graph, you will have an affine, a line, okay? And so what this means, that means that regardless what happens at large scale, low scale doesn't matter. Regardless, this will dominate. So that means that in our population, and in each condition we have about 900 to 1,000 cells. So that means that few cells will dictate the whole dynamic of the remaining cells. That's what exactly it means. So you have a few cells will direct the fate of a whole population. And we looked at distribution, and sure enough, XGB was able to rescue the distributions of the cell at the cell population level. So no mitochondria. Now we are looking at another level of mitochondrial influence. We were looking at the cell autonomous inside the cell, but now we're looking at what mitochondria can do beyond the cell at the cell population level. What you can see that XGB successfully rescued the H2AZ, improved the AZH2, and successfully restored the nuclear area, which means the morphometrics of the nucleus. And so, and to confirm that statistically, so what you need to use, you use a Kolmogorov-Smirnov test, which is a non-parametric test that actually tests for the distribution. And this test is basically a bootstrap, or you can take, which means it's Monte Carlo. So you take all these distributions, you put them together, and you start to bootstrap from them, okay? And you do that for 1,000 times, like we did here, 1,000 times. And then you start to see the distance between each of your distribution and the bootstrap, the theoretical distribution, and how close and how far it is. And then you can decide whether two distributions are coming, so you can say if these two conditions are coming from the same distribution or not. And as you can see here, the distance between, so you can see here, this is naive reserve cell versus primed reserve cell. So there's a certain distance here. And we have two values. We have a distance, which is 009. We call it the Kolmogorov-Smirnov distance, and the p-value, which is 001. So the higher the p-value, it means that the cell, the lower the p-value, the threshold is 005, of course. So the lower the p-value, it means that the cells are coming from two different distributions. The higher the value, this means the cells are for the same distribution. And so, sure enough, arsenic, the primed progeny and the naive progeny are not following the same distribution. But that distance, when you treat the cell with XGB, that distance is like 9-fold less. So you reduce them, and actually you increase the p-value to 0.8. So you actually establish the cell population dynamic. So you establish the distribution of H2AZ at the cell population dynamic by acting on mitochondria. And you basically have no effect. So it's very beneficial because you don't affect the control that you add XGB to. There is no real bad effect on it. So it's the same distribution. And we were successful in doing the same thing with area, with width, and with length of the nucleus, which are very important. There is an emerging literature that shows that the morphometric attributes of nucleus affect the genotype and the transcriptional identity of the cell. So any movement, any physical movement in the nucleus will affect the configuration of epigenetic modifier with regard to DNA or histone, and then affect the gene expression. So we were able to establish that as well. And we were able also to improve EZH2. And just to give you an idea about the methodology, so what I did is that I wrote little code just to isolate every nucleus and actually go nucleus by nucleus, so I can make sure that I'm dealing only with nucleated cells. I'm not dealing with some fused cells or some fragmented nucleus or something like that. So we use the nucleus that's as good as they can be, okay? And then we measure the area, length, width, circularity, and the intensity. And you can see that we are able to rescue, with the help of XGB, other morphometric characteristics of the nucleus. So doing that, so realizing that distribution actually obey the extreme value theory, then the next step, of course, is to try to see if we have a power law in our system. And so to do that, of course, you need to fit the cells. And so that's what we did. And I'm hoping to be able to zoom. Okay. I will zoom only on one example because I think that's the most important thing. Okay. So this is the distribution here, nuclear area, and here I believe H2AZ. Okay. So we do distribution. So what we did, so there is a lot of people who report power law in all fields, but 98% of these power laws are actually not true, because the only evidence they show is the line. Turns out that even a log normal can give you that line. Even another distribution called Weibel can give you that line. And so what we did is we tried to do it the right way. And the right way was put forward by 3 physicists from Santa Fe Institute, who put actually a principled way of proving a power law in a system. So there is a package in R language which I learned, and I applied to my data. And when you apply that package, there is a systematic approach, step by step, from defining, for example, we know that power law is asymptotic by its nature. That means that power law is always restricted to the tail of a distribution. So one problem is to define that X minimum. And to do that, you need to go through bootstrapping test and all that. So we did all that. Bottom line, we used a very principled way that is widely accepted as the gold standard in terms of proving, statistically talking, the power law. And what we found out is that the tail of our distribution is a power law. And actually, most of them are pure power law. They are Paretos, which is the purest form of power law. So we did all that, and we found this distribution. And this is just the methodology, as I mentioned to you. So you do a bootstrap again, it's a Monte Carlo simulation, we simulate a number, and then we define the X minimum, and we define the length of the tail and other parameters. And based on this simulation, we can start to actually compare our distribution to some standard distribution and see whether we really have a power law in our system. And so we did that, and then, as you can see, this is the distribution of area. I believe this is area. Yeah, this is area, this is H2AZ, this is H2. And you can see that they all have, I'm going to zoom in on some example here. So you can see, this is the simulation, the power law, and this is the empirical data, which is my data. And then you can see that there is the package actually lets you decide what is the degree of power law you have. And we can get lots of metrics, and unfortunately the image is not that great, but we can get the goodness of faith, the X minimum, the p-value, and all that. And what we found out is that we have so the tail of our distribution, the tail of distribution of H2AZ, EZH2, and the morphological nuclear, the nucleus are all obeying a power law at some extent. But the cool thing is that we are able to, by intervening with XGB, to affect that power. Okay, so because of the asymptotic nature of power law, that means that power law is always restricted to the tail of the distribution, to that 20% or 1% of the population who control everything. So then, the next approach, let me back up here again. I forgot to say something. Okay. So when I fitted the whole data, I choose the 3 top fitting distribution. So you can see here, I'm putting 3 something here. I mean, if you can see close, but I put the 3 top distribution. And what I noticed, the pattern that emerged from this fitting is that either I have extreme value theory distribution or I have mixture distributions. Turns out that the mixture distribution are actually generative mechanisms of power law. So the way that works is that when you have, in some population, it's very heterogeneous population, you have a superposition of different subpopulation with different mean and different standard deviation. The superposition of that actually generates a power law in the system. So it's actually a reflection of a heterogeneity of the system. And then we truncated that, and then we did something called survivor probability, also called reliance probability by engineers, which basically just give you the probability of an event to happen given a threshold. And as you can see here, if we look at the tail, the probability of a reserve cell, that's a reserve cell progeny that was primed by arsenic, to have a big area, or to have a big amount of H2AZ, or to have a big amount of AZH2 is much higher. So what we found out is that the decrease of probability in all the conditions that were either control or XGB plus XGB intervention decreased exponentially, while the primed cell actually decreased algebraically. So that means you have much more probabilities to happen here. And when we, of course, plot this on a log-log plot, we were able to reproduce exactly what power law would tell us to expect, which is an affine. Okay, so this, from this we can actually learn, we can have a lot of insight, and I'm just going to give you a couple of points of contacts. Number one, this data tell us that if there is a mechanism that is mitochondrially based, it's not one mechanism. It cannot be one mechanism because you don't have one state. You have a distribution of states. Each of these cells, each of these nucleus, because ultimately the phenotype of the cell is determined by its protein, basically. So the protein is the protein that defines the phenotype of a cell. And what we see is a great variability in terms of protein distribution. So that tells us that there is not only one single mechanism that is dictating that. There is so many mechanisms. So then what's going on? So then we go back to the initial hypothesis that I put forward, which says that mitochondria may be actually some integrative platform of different mechanisms at different level. In this case, mitochondria may be, okay, on one hand, controlling proliferation, controlling self-renewal, okay, but on the other hand, controlling also the distribution of H2AZ, AZH2, and other metrics at the cell population level. In fact, what the imaging data, and I don't know if you noticed that, my imaging data I show you, you can see that mitochondria reach out beyond their normal limits of a cell. And that's a phenomenon that I actually noticed like 4 years ago, and I discussed with Dr. Cheikhi when I was doing the quantification with MEX and HeLa cells, and I reported to you that I saw mitochondria jumping from cell to cell, and it was kind of weird. We didn't understand what's going on. But now we know now that mitochondria have long-range properties. Mitochondria, because of these nanotubes that can form between cells, what you can have is basically a whole package of mitochondria with all its program going from cell to cell and then imposing the program on the nucleus. Because let's not forget, mitochondria is the source of, so you can think of it that way. So the way I look at it, at least, the way I look at it is that the DNA is just the hardware, okay? And sorry, the DNA is the hardware and software. But for that code to run, you need an input. The input is mitochondria. The input can be acetyl-CoA, can be some other metabolites, can be H2 hydrogen peroxide that will modify some protein or something like that, or some provoke some methylation or something like that. It can be cardiolipin language, something coming from mitochondria that carry information. That information is the input for the software to run. So that's the way I look at it. Okay, so in order to prove, to provide a proof of principle that this idea might be correct, we actually looked at we did some epigenetic studies to see if that's correct. And what we find out is that, so we looked at two things. We looked first at the demethylase 1 and 2 and 3. So we know that the DNA demethylase 1 is responsible for static memory. So that's you have a whole template that is going from mothers to daughter cells. And we know that the DNA demethylase 3A is de novo methylation, and that's responsible for dynamic memory. Okay? And of course, we know that H2AZ, especially when it gets acetylated, it actually will define the bivalent chromatin marks. So this is areas of DNA that are poised to be either way. Okay, so they can go differentiation or they can go back to self-renewal. And that maybe explain why you have just amount of mitochondrial work provided, because to maintain that state. So it's like you are planning to travel to the North Pole, to the North Pole and to Africa at the same time, and you are preparing for both. So it's a lot of planning, because you need to go to different environments. So, commit to differentiation or go to self-renewal. And so you need a lot of work and a lot of energy. And perhaps that's the reason why we see that high mitochondrial profile in terms of potential and all that. But again, the cool thing is that we use, and by the way, I'm using XGB at 100 nanomolar. Okay, this is very low dose of XGB. And the reason why I used 100 nanomolar, because I wanted to see which performed best, XGB or SS31. And I compared both. And in my hand, I found XGB superior to SS31 at that concentration. Now, keep in mind that SS31 was reported to also operate at 10 nanomolar and be effective. Some people even claim to use it at 1 nanomolar, and I don't know if that's true, but that has been reported as well. But in our hand, we know that at 10 nanomolar, SS31 is a little bit kind of doing better, but at 100 nanomolar, definitely XGB is doing better, and most importantly, we get the effect, the desired effect we want, which is the reversal. And so when we add XGB, we were able to rescue both the static and the dynamic memory, and we were able also to change the post-transfer modification of H2AZ, which control thousands of genes, and the whole cellular memory, actually. Not only that, but as you can see from the mRNA level, we were also able to control the RNA polymerase II. Okay, so the picture that emerged from this is that mitochondria is actually optimizing. So that's the word optimized counts. So optimized, so mitochondria is affecting many processes that are somehow correlated at certain level. Okay, they may be epigenetically correlated at the cell population level, other processes, and other level. So that's what we know for now. And by optimizing these processes, XGB is able actually to rescue the phenotype. So we conclude that that mitochondria is causally linked because we used a drug that is targeted to mitochondria we proved and we demonstrated that we were able to modify pretty much every parameter of mitochondria, and then the outcomes have necessary to be derived from that. And so we know also that XGB is I think 1,000 times more in mitochondria. So, and given the very low dose I use, we are almost certain that this conclusion is correct. And so we know that now we know this also. This is the first time reported. We know that mitochondria now can control cellular memory actually. The distribution of epigenetic regulator that include the morphometric. So this is also, I don't think was reported before. So we report for the first time an intimate relationship between morphology of mitochondria and morphology of the nucleus. And so what this, the significance of this? So for the cell to make decisions, the cell must be able to somehow relate past resolution, past states with future decisions. Okay? If something affects the memory of the cell will not be able to make that kind of linkage and then cannot make the right decisions. So under hyperfused mitochondrial regime, what you have is you converge in one state. So you have hyperfused state and you have very constant activity of mitochondria that actually push the system to converge to a certain number of states. So it reduces the number of states that the system can take. On the other hand, so that's in Kaufman terms, that's what you would call an ordered regime, okay? Now, if it is fragmented, that's what you would call a chaotic machine, so this chaos. And in chaos, of course, you have this, even tiny, small noise can actually give you very unexpected outcome. So it's not good either. So nature favors something in between, something that is complex, something that is a marriage between order and chaos, yet favorable for life and favorable for healthy choices for the cell. And that regime is a balance between fusion and fission. So you have a certain optimized state of mitochondrial network that supports certain operation at the epigenetic level to happen. Okay, so for future directions, I'd like to have the opportunity to understand more the role of mitochondria, the interface of extrinsic and intrinsic cellular processes, and this is an area that I don't think nobody worked on that before. Although there is a lot of reviews that point to that, but there is no work that has been done to that regard. And another thing that's actually very interesting is the mitochondrial shape per se. So shape is information per se. And recently it has been shown that there was some experiment where people actually forced the cell to be shaped a certain way, and based on that they can actually dictate what kind of chemical reaction happen and what kind of receptors will bind or not. And so it's a very interesting idea to think of shape as some kind of integrative mechanism. Of course, when we talk of power law, we talk about what we call self-critical state. It means that somehow the interaction of the components of a system will emerge and give you something that is independent of the microscopic details of that system. So that's what it means. And that's why the power law is so fascinating, because it gives you that, it leads to the hypothesis that somehow there is that, universal mechanism that can explain a lot of complexity we see in the world in many areas. I would like also to continue work on these mathematical and computational tools that the physicists and mathematicians and computer scientists came up with, and I think this is a very good thing. I think that something will help us. I'm not trying to be a mathematician or computer scientist. I'm just trying to learn how to use them properly and use these tools to have actually a biological insight for myself and understand more about the cell. The last thing I would like to say is about cardiolipin's language. And the reason why I would like to bring that up is because it turns out that H2AZ control oleic acid. So more H2AZ, more oleic acid in the system. And I know that Dr. Ciurina published a paper when they feed, I think, oleic acid conjugated to something, and they give it to some cells to mix. And the result of that was when cardiolipin got enriched in oleic acid, it became resistant to that. So I'm suspecting that there is some kind of, based on this just preliminary data I have on cardiolipin, I'm suspecting that that's part of the resistance to that. So you have this huge activation of caspase, yet you don't have any cell death. And you have some oxidation of cardiolipin based on the NRO. So I'm suspecting that there is maybe something going on with the speciation of cardiolipin that actually enable the whole mitochondria to act that way. And so it will be interesting to understand how the cardiolipin language actually coordinates structure of mitochondria, morphology, energetic, the bioenergetics of mitochondria, and the process of information that is actually given to the nucleus to decide the fate of the cell. And that was it. Thank you very much.
We have some time for one or two questions.
So I mean, I'm perplexed that so that you're flying so high, and you do modeling, mathematical, computational. So, and I still stay grounded to very small and miserable things like individual molecules, what affects what. I don't have this very interesting view that you have. And actually, it's kind of interesting that listening to you, I was thinking about also Forrest Gump when he started running. Forrest, why are you running? I don't know, I just like to run. So, what's the analogy? Well, in the end, you have two compounds. One is arsenic, and another one is XJB.
Yes.
Right? And both of them are doing something. And forget about modeling for a second, because this is statistics, and yes, you can find 2-3 individual bright people or bright cells that are mostly affecting the behavior of the system. That's interesting, of course. But in these two individuals, 2 cells or 2 people, arsenic targets something, right? Because that's with 20 nanomolar or any concentration, but there's a molecule or 2, or you suggest that there are many, more than 2,
Yeah.
several, but there's still few molecules, and they trigger this all. And now you go to level 2, 1, 2, 3, higher hierarchies of this, and find very interesting interactions.
Mhm.
But there's still the beginning, and I am always, with my very grounded view, I'm always interested in the beginning.
Okay.
What is it? What is it, and where is it? Is it in mitochondria, or it's not in mitochondria? XGB acts in mitochondria, but does arsenic act in mitochondria?
No, not necessarily.
No, I'm asking still.
Okay.
So, because there's a simple experiment. Kind of, you started with showing different thiols, protein and others, GSH, and everything goes down. Well, is it the prime target? We've been talking with Aaron this for centuries now. So what is arsenic doing? Does it affect thiols somewhere? Where's the beginning? Where in this power law obeying cells, what is the target?
Okay.
Without doing this, yes, you can do, and I appreciate the effort, and I think it's spectacular. But where's the beginning?
Well, so that's the idea. That's precisely the idea of thinking of mitochondria. So when you have a proliferation like that, when we put in this system, so what you have is far from equilibrium system with it's stochastic, okay? And so this is actually a very complex dynamic system. And the idea is precisely what you just said. It's very hard to pinpoint that particular target you're talking about. So in order to move on with the understanding, you try to approach things at much higher level. Now, I totally agree with you, and I submit totally, and you're right, 100%. And I have some idea how that might start. Okay, so the response of glutathione, for example. So that increase in glutathione tell me, for example, that the glycolysis should be up. Okay, so we know that, and we know also so we know that arsenic actually induces the Warburg effect, which means you have a very high glycolysis, and you will have very high mitochondrial potential, exactly like cancer cells. However, the cell, the mitochondria in this kind of cells actually doesn't produce ATP. It's actually consume ATP. And the reason why it's consume ATP, because you need to deplete the cytoplasm from ATP to keep the glycolysis going, to keep your proliferation going.
What is the prime target? Because now you're going in the same direction.
Probably I would say probably tiles, probably that can be. I don't know exactly, to be honest. I don't know. I thought about it in the same way you just said.
You know that you are a co-author on a paper that we published with you.
Yeah.
And that was a paper that described this approach of optolipidomics.
Yes.
So why did we go for it? Well, because we have had this exactly same issue. And we used XJB like you can. Well, what is going on? You prime cells to apoptosis by doing one very simple thing. We used the same non-anilacridine orange that you have used.
Yes.
And you started it, remember?
Yeah.
You were shining light on this non-anilacridine orange, and
Yes.
healing these cells by very, very minute doses of radiation, but by oxidizing these cardiolipins. So why did we do it? Well, because we had this mountain of information before, and you quoted paper by Hewley and Gingy. Well, there's apoptosis, cardiolipin gets oxidized, and with the root mass spectrometry, we identified 200 species of cardiolipins that were triggered by this. Okay, which of them are signals of apoptosis? Who knows? But by using optolipidomics, we identified only 3 out of 150. Why did we do it? Well, because we wanted to know what is the prime target. So there are 200 signals, but 197 of them, they don't signal apoptosis. They signal something else. Only 3 are important as signals for apoptosis. Okay?
Yeah.
Similarly here, I think it may be in a similar way. So there may be many, many, many signals that are generated. And yes, you see decreased amounts of cardiolipin, and something else goes up and down. Many of them are consequences. What are the initial signals? That's what you, in my, again, very
I think
grounded.
it's going to be we actually have to get out of the room, but no, it's a great comment. We're still searching.
I'll come in 3 years.
Thank you very much.
Thank you.
The committee members will be convening downstairs.
That was very interesting.
Thank you. Thank you.
Big problem you were told.
Yeah.
Someone was